Today's lecture is going to be on the second aspect of chapter 14, the nerve structure. When we're looking at a nerve and the general connective tissues, we're going to be looking particularly at how a nerve is a cable-like bundle where we see multiple axons running in parallel with each other. Just like the muscle tissue, the nerve tissue has three successive connective tissues. The outermost, or the epineurum, the perineurium, and then the endoneurium. The first one we'll begin the discussion is going to be the deepest layer, which is going to be called the endoneurium. This endoneurium contains a single axon, and of course it is going to be encased in a myelin sheath, which is going to be surrounded by neural lemmacytes, and then each one of these individual axons will be wrapped in the connective tissue of the endoneurium. Moving further out, before we get to that, then what we see in this layer also is a thin layer, this white substance right here, a delicate layer of areolar connective tissue that helps separates and electrically isolates each one of these individual axons. Also within this connective tissue, it's not pictured here, but we have a series of capillaries that are going to help supply each one of these individual axons. Moving out the perineurium, we see here we have a group of axons wrapped into separated bundles, and these separated bundles over here are going to be called a fascicule, very similar to the muscle tissue. And they are going to be covered by this outer layer located here and also here. It's another illustration by a dense irregular connective tissue called the perineurium. This layer also helps support the blood vessels. The blood vessels, again, are going to supply the capillaries, which are located in the endoneurium. And moving on finally to the outermost layer, the epineurium. This is the most superficial connective tissue. This thick layer of dense, irregular connective tissue encloses the entire nerve, providing both support and protection to the individual fascicular bundles. So when we start getting into synapses, the first is to look at the general definition of a synapse. These are specialized sites where a neuron communicates with another cell. In this particular example, we're going to talk about the difference between the presynaptic neuron and the postsynaptic neuron. So in the presynaptic neuron, what we see is the presynaptic neuron will transmit nerve impulses through their individual axons towards a synapse. And the synapse is going to be this region that we see between the presynaptic and postsynaptic neuron. This synapse is also going to include the synaptic knob and the small separation between the synaptic knobs of the presynaptic and postsynaptic neurons, and that's called a synaptic cleft. In the postsynaptic neuron, what we see is that it's going to conduct the nerve impulse, as this arrow shows, through the dendrites. So once the synapse occurs in the synaptic knob of the presynaptic neuron, it will communicate or send the nerve impulse through the dendrites processed by the cell body, and then it will then send the nerve impulse away from the original synapse. So down here we can see just a general overview. We have the cell body, the direction of the presynaptic neuron will be sending the synapse or the nerve impulse towards the synapse. The synapse is composed of the synaptic knob and the synaptic cleft. It's picked up by the dendrites of the postsynaptic neuron, and then that impulse will travel down the axon away from the synapse. When we're looking at the three common types of synapses, the first one we'll look at will be the axodendritic synapse. So the axodendritic synapse is the most common. It occurs between the synaptic knob of a presynaptic neuron and the dendrites of the postsynaptic neuron. So here we see this would be the pre and this would be the post. This was the same illustration that we just went over in the previous slide. The axosomatic, this one right here, we can see a little bit of a different structural makeup. It's going to occur between the synaptic knob and the cell body of the postsynaptic neuron. So the presynaptic neuron is going to have the synaptic knob, and the postsynaptic neuron in this example of an axosomatic will be the cell body. And finally, the last one is the axoaxonic synapse. And this axoaxonic is the least common synapse. And what we see here in this example is the synaptic knob of a presynaptic neuron and the synaptic knob of a postsynaptic neuron. So we have synaptic knob of a pre and a synaptic knob of a post communicating with each other. This action appears to influence the activity of the synaptic knobs. There's two different types of synaptic communication. The first one will come over here and we'll talk about the electrical synapse. This is where we see an electrical synapse. We're going to be dealing with plasma membranes. So here on the postsynaptic cell, we have the plasma membrane, and the presynaptic cell, we have a plasma membrane. And the plasma membranes of the pre and the postsynaptic cells are bound together. And they're going to be bound together by these structures right here. These are the connexons. So we can see one here and one here. And we learned earlier in chapter 4 that connexons have a pore. So with this, it helps facilitate the flow of this example is showing sodium ions that are going to flow through the pore of the connexons that are joining the presynaptic and postsynaptic cell. So in this illustration, we can see as the local current flow, this voltage change is going to cause movement of the charged sodium ions to transfer from the pre to the post, which will then allow for the nerve impulse to propagate from the pre to the postsynaptic cell. This is important because with this connexon and pore and ion exchange, the nerve impulse passes between them with absolutely no delay. We see these primarily occurring in smooth muscle cells in the intestines, and we also see this makeup in the intercalated disc of cardiac muscle. And then finally, the most interactions between neurons and all communication between neurons and effectors are going to be this chemical synapse. So here we see the direction of the nerve impulse is coming towards, and this presynaptic neuron is going to be coming towards the synaptic knob. So the first thing we see is that the nerve impulse travels the axon towards the synaptic knob. Once the arrival of the nerve impulse, this allows for calcium ions to move into the synaptic knob, and these are moving through these voltage regulated calcium channels. So the only way calcium will move into the synaptic knob is first with the initiation of the nerve impulse. once we see the calcium coming into the synaptic knob, this will then trigger the synaptic vesicles to move distally towards um, the plasma membrane of the synaptic knob, and it will allow for the neurotransmitter of acetylcholine to diffuse into the synaptic cleft. So the neurotransmitters are then going to attach um, into the postsynaptic membrane, and the influence of the acetylcholine is then going to allow the influx of sodium to come into the postsynaptic cell. And then what we see is the charge or the nerve impulse will then propagate along the postsynaptic membrane. Once this occurs, then we see an enzyme called acetylcholinesterase, which will help break down the acetylcholine. So again, as the nerve impulse comes in, it causes these voltage regulated calcium channels to open. Calcium comes into the synaptic knob, causes the synaptic vesicles to move towards the plasma membrane of the synaptic knob, releasing acetylcholine. These acetylcholine will then bind to receptor protein, causing these ion gates to open. Once sodium crosses into the postsynaptic membrane, then we see the action potential propagating across the entire length of the postsynaptic cell. And then the acetylcholinesterase will help break down acetylcholine to allow for another nerve impulse to travel through and to repeat the cycle. And finally, when we're looking at neuronal pools, these are defined strictly by their function, so not their shape, but what is going to be the final output. So the first one we see here is we're going to have multiple inputs. This is called a converging circuit, which means basically converging means to come together at a single individual postsynaptic neuron. We see this in the salivary glands. We also see this in hearing. So basically, for the production of saliva, maybe the input over here would be the smell. The input here is coming from the optic side, which means that you're seeing the food. Maybe you hear the food being prepared, and also communication about the food with the people in the room. And all of those multiple inputs are going to converge, which will then cause the salivary glands to release saliva. The next one is the diverging. This one is going to spread information. So we see information from one presynaptic neuron is going to then multiply into multiple outputs on the postsynaptic neurons. We see this in walking. For instance, just the general input of walking across the classroom will then cause, the activation of the sensory of your feet, the activation of the vision of walking towards the door, and also, the tactile nature of what you're walking across, and we'll see multiple outputs. The multiple outputs would be moving your left leg, moving your right leg, moving your left arm, moving your right arm, turning your head to make sure that you walk correctly through the door. And all that was stimulated from the initiation of the voluntary control of the simple movement of walking. The reverberating circuit, this utilizes a feedback. So we can see here, this feedback allows for repeated cyclical stimulation of the circuit. Once this is activated, the circuit will continue until there is an inhibitory stimuli or synaptic fatigue. So this is what we see with breathing at night. So for instance, the general act of breathing, so inspiration and expiration, is going to produce the movement of the lungs. So at the postsynaptic membrane is going to produce inspiration and expiration. And then the multiple inputs that will be occurring would be maybe the moisture in the air or the partial pressure in your lungs. And all of these are going to continue to work in a circulating pattern until there's a disruption like an inhibitory stimulus or a synaptic fatigue. And finally, the last one we see is the parallel after discharge. Parallel after discharge is really interesting. You can look at this as a critical thinking. We have several neurons that are processing the exact same information at one time. So we'll see a presynaptic neuron here stimulating different groups of other presynaptic neurons, passing that nerve impulse, that synapse with a common output. We see this in critical thinking, such as higher order of thinking, such as a mathematical problem. So taking, A plus B plus C and finally figuring out what the answer would be. Or a biological test, understanding each one of the scientific terminology and then allowing the brain in a higher order of thinking to communicate the answer for the test.